So everybody, thank you so much for coming tonight to Strait Science. Strait Science is an evening science presentation put on by UAF, Alaska Sea Grant. This is the home office, and also UAF Northwest Campus in Nome. And together we serve both Alaska Sea Grant and Northwest Campus. We serve the people of the Bering Strait region, which is the homeland and waters of the Yupik, Inupiaq, and St. Lawrence Island Yupik peoples. And tonight we welcome Kathi Lefebvre. Her topic is going to be what happened in the warm year of 2019. And she is studying Arctic food webs and harmful algal blooms and harmful algal toxins, which is a topic that we need more information on, that is for sure. And we're going to get some of it. And she is a leading expert, and she is doing a project that is right here going on along Western Alaska, but certainly in the Bering Strait region with repercussions and information for all of us. Kathi is a research biologist at NOAA's Northwest Fisheries Science Center, and she's calling in tonight from Seattle, so she's on a different time zone as well, and we thank her for staying late already. And she's also the leader of the NOAA's Wildlife Algal Research and Response Center. So she is no stranger to talking about saxotoxin or any of the harmful algal toxins when it comes to wildlife. And that is really important for this region, especially we're talking marine wildlife. So with that, Kathi, I'm going to hand it off to you. Oh, wait a minute, before I do that. We have callers on the line. So callers on a Strait Science presentation get priority, because if you're calling in, you are not able to see the screen. You can't raise your hand, and so you will get priority. So for the callers calling in from St. Lawrence Island tonight, know that at any time, please speak up or start your question, and we will stop for you. And also you have my cell phone number, so call that if I'm asleep at the switch. We're still getting people coming in, so that's great. I better turn off that doorbell or I'll drive Kathi bonker, all of us bonkers. And with that, Kathi, we are all ears on this important topic. Thank you so much for coming.
All right, great. Thank you, Gay. Okay, I am excited to share with you what we're learning about algal toxins in Arctic food webs and the potential exposure of these toxins to walruses and bowhead whales, which of course is very important resources in this region. So just to sort of start out, I'm going to give okay, I'm trying to advance my slide. There we go. Okay. So just to give a sort of introduction to what I'm talking about, what do I mean by algal toxins? So I'm going to be talking today specifically about paralytic shellfish toxins. We also call that saxitoxin. That's the main toxin we think of with paralytic shellfish toxins. And as many of you already know, these are produced naturally by single-celled microscopic algae in the ocean. And this little schematic here is courtesy of Don Anderson at the Woods Hole Oceanographic Institution, and it shows the life cycle of the species that we're concerned about right now, and that's Alexandrium Catnella. If you look to the right of the panel, you can see these two round cells. Those are the vegetative cells, they're called, and they just live in the water column, and when the conditions are right, they can divide and multiply. And when they multiply in big amounts, we call that a bloom. And because they produce these toxins, we call them harmful algal blooms. Now, the interesting thing about this algae species is that it also has a cyst stage. And you can think of the cyst stage as like a seed. These vegetative cells can create this resistant cyst that goes down and sinks and sits in the sediments and stays at the bottom of the ocean. And it just stays there until conditions are right. And then it can germinate and create more of these vegetative cells that come up into the water column and cause a bloom. So that's sort of the background of what we're talking about. Now, when we have these blooms of algae and they're producing saxitoxin, which is a toxin that affects nerves, it can move through the food web because these things are eating the algae. And what we're mostly concerned about is as it moves through the food web, things like higher predators like birds, mammals, humans can become ill after eating contaminated foods. And when it's an event with humans, it's called paralytic shellfish poisoning. And I think probably most people on here have already heard this. This is just a quick review, but this happens because saxitoxin blocks nerve function and basically can cause paralysis or breathing difficulties and even death in the worst case.
I've got a question, Kathi.
Okay.
Right off the bat, and that is, I appreciate this jumping right on the saxitoxin, but a lot of people don't have a review or don't know about saxitoxin, and I'm always learning myself. And so saxitoxin is just one of, or this Alexandrium is just one of all different kinds of algae, but we have lots of good algae in the ocean in this area. Is that, I mean, this is just one kind, right?
Yep. No, that's true. Yep. There's a lot of, well, yeah. So phytoplankton or algae are very important. They're the base of the food web. They help with the atmosphere and the environment. And there's a lot of good algae. This particular one that we're talking about, Alexandrium, is one that produces these toxins, and that's why it's concerned, and that's why it's called harmful algae. But absolutely, not all algae is called harmful algae. That's a good question.
Yeah, and so we're not, it's not new to the region. I mean, Alexandrium isn't new.
Nope. And yep,
right?
I will definitely, Alexandrium has been here probably for hundreds of years. So that leads right into this question. Well, if we've had Alexandrium, if we've had Alexandrium in the region for long periods of time, why are we concerned about it now? And that's what I'm going to share with you in the next couple slides, is another sort of review. And if some of you were at the Strait Science seminar that Don Anderson from Woods Hole Oceanographic Institution gave a few weeks ago, I'm going to sort of review some of that because that lays the groundwork for what I'm talking about toxins actually in the food web part. So this is a graph actually showing some data from Don's lab where they could take these Alexandrium from different locations in the ocean, in the world, bring them back to the laboratory, and then they can grow them in the laboratory under different conditions. And one of the things they're finding that really influences the growth rate, that is how fast this algae will grow, is temperature. So that's what this graph is showing here. As you increase the water temperature, you get a faster growth rate of those Alexandrium cells. They multiply faster and therefore can create a bloom, and a larger bloom. And I want you to look right at, if you have a handout that isn't in color, you can't really see this, but one of those lines that's on this graph is actually Alexandrium isolated from the Chukchi Sea and grown in the laboratory to show this. And if you look at that bar that's on the graph with the arrow pointing down at it, that tells you the temperature in the Chukchi Sea at the time they isolated, or that at that time period. So really what this graph is showing you is that as water temperature warms, growth rate increases for this Alexandrium species. I think you can tell where I'm going with this, why this is now a concern in the Arctic. And this is one of those great graphs that Rick Thoman always has and can show these long-term data sets. This is showing average sea surface temperature in the northern Bering Sea from 1900 to 2021. The only reason I'm showing this is to share with you what you guys already know. You're living there, you know this is happening. If you look at those red dots at the end, those are the 10 warmest years on record since 1900. All of those are after the year 2000. So there's this obvious concern about continued warming. So I want to expand that story just a little bit further. So now I've shown you that warmer waters will increase the growth rate of these algae species that produce these toxins. And this algae has been in the Arctic for many, many years, but now there's the potential for faster growth rates. So there's another part of the story. Remember, I talked about cysts. These guys have cysts that can go down into the sediment. So if you look at the top panel of this figure, you can see the arrows, and that's basically showing you that these cells, Alexandrium, and those are the little dots on there. If you don't have color, they probably look like black little dots. And we are saying, or we think, that these Alexandrium cells are coming from the south, moving up through into the Arctic. There's a place where it kind of slows down in the Chukchi Sea due to oceanography, and there are cysts that can then drop to the sediments. And this has been happening over hundreds of years, and the cysts have been sinking to the sediments, and they've been building up. And so if you look at the lower panel of this sort of figure, it shows the historical look at that. So cells are coming in from the northern Bering Sea, slowing down in the Chukchi Sea, dropping cysts, and those cysts are then building up in the sediments. Historically, the bottom waters have been too cold for the cysts to germinate. So that's right, you probably guessed it. The cysts also are dependent on temperature, and the warmer the temperature, potentially the higher the germination rate. So if you look at the panel all the way over to the right, that's what's happening now. The bottom temperatures are now warm enough for these cysts to germinate. So we have not only vegetative cells in the water column that are growing faster because it's warmer, we have vegetative cells coming out of cysts because they can now germinate quicker because of the warmer waters. So that's sort of setting the stage for the fact, or the idea, that there's going to be potentially more toxins in the food web during blooms because the blooms will be larger.
So we have a question in the chat box if you want to take that now, and that is, knowing that these Alexandriums are one type of plant cell, algae, and amongst a whole host of different kinds of algal types that aren't harmful, is there, with these warming waters, and this Alexandrium seems to like warmer water, is there a temperature range that could kill that toxic algae? Or do they just...
That's a great question. And actually, I should have kind of pointed that out here. If you look at this graph, I'm going back a slide. If you have the handout, I'm going back.
So this is the handout that has at the top, why are we concerned about the Arctic, on that page?
If you look at that, you see those lines that are, you see the temperature going up, and you see those lines of growth rates for the algae. And you see that at the end, at like 22, it says they're dropping off. So yes, you are absolutely right. There will be a temperature that is too warm for these algae. That's an excellent question. However, that's really a long ways off. Okay, so now I want to tell you what we're doing to look, or what we did in 2019 to look at toxins, these toxins in the food web part. So how much toxin are we seeing in the different parts of the food web? So I'm going to share with you the data that we got in 2019 from these massive collaborative effort cruises, so this figure here shows you all of the sampling sites that we got samples at. We sampled for zooplankton, clams, worms, and fish. And then we measured toxin levels in all those organisms from all the different locations. So the next one, this is just another great graph from Rick, just to remind you what the conditions were like in 2019. This figure here shows sea surface temperature departure from normal. I believe that 2019 is still the warmest year on record for water temperature. And so let me show you what we found. So we'll start with zooplankton. So if you're looking at the handout, it says zooplankton, and there's a map to the left. And this is where we sampled krill and copepods, and we took a scoop of zooplankton, and we measured saxotoxin levels in those. And I'm going to show you a series of these graphs for the different organisms. And so I want to explain this scale for you. And if you don't have color, I'm going to have to just describe some of it. But what we've done is we are relating the toxin concentrations that we found in the zooplankton to the seafood safety regulatory limit set for shellfish. So the seafood safety regulatory limit is 80 micrograms saxitoxin equivalents per 100 grams shellfish. So the scale that we created is, if you see white, that means we didn't detect toxins. So if you're looking at those little animal icons on there, if it's white, we did not detect toxins. If it's yellow, we had low levels of toxins. Those are toxins below half the regulatory limit. If they're orange, it's at the half the regulatory limit up to the regulatory limit. And if it's red, it means it's at or above the seafood safety regulatory limit. Now, if you don't have color here, I just want to say that most of these were low levels. There's a couple orange, which would be moderate. And there was one near Ledyard Bay that was above seafood safety regulatory limits for zooplankton. The next slide that I'm going to show you has clams and worms. So we sampled all these clams and worms from all these different places, measured for toxin. And what you can see right away is that there's more toxin in clams and worms compared to zooplankton. And we have the same scale on here. Mostly there's a lot of low, there's a lot of moderate, and then there are 3 places, one in the Bering Strait, one near Ledyard Bay, and one up near Utqiagvik, where we have clams taken from the bottom of the ocean that had saxitoxin levels above seafood safety regulatory limits. So if those were harvested for human consumption, they would not be safe to eat. They would not be considered safe to eat. So that was surprising to us. These areas have not really been sampled before, and I did not necessarily expect there to be high levels of toxins in these deep water clams in these locations. So that was a surprise. Another thing, we're going to try to be modeling where these toxins go and how much or animals like bowhead whales or walruses can consume and when they might have health impacts. So one interesting phenomena that we're finding is different parts of the food web have different concentrations of toxin. And in this case, clams versus worms, 100% of the time, in every situation where you get a clam and a worm at the same time and location, the clams will have higher toxin concentrations per gram than worms. So it's something that's interesting. The next thing we're going to show here is the fish that we looked at this year. Now, this was sort of surprising to me because I do know that fish can accumulate toxins, but what we found in 2019, very,
What
very.
about sea peaches? Hello?
Yes,
Go ahead.
is that good?
Can you hear me?
Yep.
Yes.
We missed
I
your question.
didn't see no sea peaches out here because we eat sea peaches out here.
That is
See-
one of the,
And you don't,
go ahead.
oh, go ahead.
You are right, Ben. We did not look at sea peaches. Are those
So
cucumbers?
So if I can answer that one a little bit. So yes, she was unable to look at the for the science world, they'd call it a tunicate or a sea peach, but actually that is what she's still looking for. So if you do want to turn in, and we'll talk about that at the end, I think, but if people want to contribute to this food web study that she's doing, it's a research project, so it takes a while before the answers come back. Like this is 2019, and here we are in 22. But that happens. But absolutely, I mean, getting hands on for her to be able to look inside UPA and be able to determine if it has saxitoxin in it and at what level. Absolutely. If you can send those in to me or Tiffany, I think is Tiffany Rocock, if this is Savunga. I think she's your environmental officer now for the tribe. So she would be the one to hand it and she'll get it into NOAA, if that helps.
Yeah, thank you.
You bet.
Hey, this is Ben.
Mhm.
I was looking at the that food web I see just human on the. There are scavengers like fox out here. I've
Yeah.
seen a dead fox for some drift seafood that washed in. Every once in a while, when I go to the seal blinds, I see this ferocious mammal come out to do some beach combing too. Not longer than my thumb, but the shrews.
Okay.
Yeah. Well, and actually, all mammals will be impacted by the toxin. So if there are terrestrial animals that are eating the toxin, they can absolutely be impacted.
So I,
All right.
Okay. Thank you.
Yeah, thanks, Ben. Okay, so just to finish up here with the fish part, this year in 2019, we saw very low levels of toxin in fish. That doesn't mean that'll always be the case, but in this particular year and on this study, these were very low levels in fish. And in fact, what I've mapped here are toxin levels in the guts of fish only, not whole fish. And most toxin in a fish will be in the gut, probably 90% or more. So if you were to equate this out to a whole fish, these levels would be even lower. So that was interesting to us. We were expecting that there might be similar to clams or something. So okay, so now let's get to what we're really interested in, and that is walrus and bowhead whales. What is the potential for exposure to these animals? So in collaboration with many of you that are on the line here from the subsistence harvest communities, we are able to get fecal samples from 9 bowhead whales in 2019 and 13 walruses. And we tested those for saxitoxin. And so just looking at this figure, I will tell you the information if you don't have the color figure. 7 of 9 bowhead whales had detectable levels of saxitoxin, but all of those levels were considered low. All 13 walruses contained detectable levels of saxitoxin, and 2 of them had moderate levels, a little bit higher. And this is in relation to the regulatory limit. Now, the question that you all want to know is, are these animals impacted? Are they having enough toxins to impact them, their health? And we cannot answer that question with just looking at fecal sample one-shot concentrations. So we don't know if these animals had high enough doses of toxins to harm them. But what we can do with this, and what we did, is we then tried to use calculations to determine how much toxin the whales and the walruses could be receiving in a 24-hour period from eating the food that they eat, because we know the toxin levels in the food that they're eating. Yes, Gay.
and this looks like a pretty important point and a pretty important slide. So for people who are following along on a printout, this is the slide that has kind of cartoon walrus and bowhead, and then big arrows pointing over to the maps that go back to the krill that were collected and the clams and worms that were collected, just to make sure everyone's on know where you are. And Dean Stockwell has his hand raised. Dean, you got a question?
Yes, I do. So even though you have sublethal concentrations in the water and in the fish, a clam can sit down and basically filter over 20 liters of water a day and accumulate those toxins. And then a walrus comes along and it doesn't eat just one of these clams, it eats many of these clams, and it can accumulate these toxins. So do you have any kind of feel for, I guess, the spin up of what these concentrations really mean? Like I didn't see a maximum concentration in your clams. You said some were at that 80 regulatory level, but some could have been a lot hotter.
That's true. And Dean, thank you. You are queuing me up. This is exactly where I'm going. So we are looking at the toxin concentrations in the clams that are food for walruses, and the toxin concentrations in the zooplankton that are food for the bowhead whales. And what we did is we figured out how much food these animals have to eat in a 24-hour period in order to do what they do, swim around and live. They have to eat a certain amount of calories. And so then we said, okay, how much food do they have to eat to meet that demand of just living? And how much toxin is in that food? And that's how we calculated a dose. And so, Dean, that's exactly what this figure or this table is showing. So if you are following along in a handout, you want to look at the page that says estimated toxin doses to walruses feeding on clams and bowhead whales feeding on zooplankton. And it's a table. And what we've done is we've taken average toxin concentrations in clams, and we've taken the maximum concentration we measured in clams. And we used both of those to calculate a daily saxitoxin dose to walruses based on how much they would eat in a day. And so the last column of this table shows you the daily saxitoxin dose in walruses eating average concentrations that we measured in clams. And that's a number of 5.2 micrograms saxitoxin equivalents per kilogram body weight. And eating maximum concentrations in clams, you get a dose of 22 micrograms saxitoxin equivalents per kilogram body weight. We did the same thing with bowhead whales feeding on krill, average concentrations of krill, maximum concentrations in krill. And then we also took the maximum concentration we saw in a zooplankton scoop sample just to see what the highest dose could potentially be to bowheads. And if you look at those doses at the end there for bowheads, they are way lower than they are for walruses. Much lower, which makes sense because the prey that the bowhead are eating have less toxins. Okay, so stay with me, Dean, because I'm going to talk more about what those doses might mean.
And just for me, for the red, the ones that are in red, the 165 in walrus, that you have that in red because the level, that cutoff level from good doses to maybe strong doses is 80. And that 165 is over the 80, and same with the 85.1.
Correct. I did color
Okay,
those red because they're above the regulatory limit. Good catch.
Okay, thank you.
Okay, so now we want to know, what does that mean? What do those doses mean? We now know, oh, that's a very realistic estimate of how much saxitoxin these animals could be eating in a 24-hour period in 2019, feeding on those prey. And so we don't know what doses cause impacts in these marine mammals yet. That's what we're going to learn. That's what we're going to find out. And that's what we're going to do as a team together through not only looking at toxin doses, but also looking for behavioral impacts or mortality events or those types of things. But what I want to do is show you, I can look back at other studies and other field poisoning events, for example, human paralytic shellfish poisoning events, laboratory models, etc., and say, hey, what doses cause impacts in other organisms? So that's what I'm showing here is this summary table. And if you are following along in your pamphlet, you're going to look for the page that has a picture of a walrus, bowhead whales, and then a big table that says toxicological metric or exposure source. And so what I want to show you is on the top 4 lines, these are information that we have from human poisonings, from paralytic shellfish poisoning. And so the center column, the wide column, shows you what the impact was, followed by the source of toxins. So for example, on line 1, that LOAEL means the lowest observable adverse effect level, one of those crazy toxicological acronyms. So this is the lowest dose in this event that caused some sort of impact in the humans. And it was from drinking water. And you can see the dose at the end there is 2.1 micrograms saxitoxin equivalents per kilogram body weight. The lowest dose to cause illness in a second poisoning event from eating shellfish was 21. And another event, a lethal respiratory arrest effect from eating shellfish, and it's a pretty high dose there, 230 to 411. But a lethal dose for humans eating crab was as low as 1 to 2. These are reported for other studies. If you look at the center, you see mouse, 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 rat. These are rodent laboratory models. These studies, you take the laboratory model and you give them different doses and you figure out when effects occur. And all I want you to see from this is an interesting phenomenon, is that those doses are much higher than the ones shown to have an impact in humans. That is not unusual. In a lot of toxicology studies, it's very well known that the larger the animal, the lower the dose per unit body weight needed for the same effect. So skip way down to where we see humpback. There's been one reported humpback poisoning event as a result we think from eating mackerel that contained saxitoxin. The dose calculated from that is 3.2. So now let's look at our doses. What did we find? What did we calculate from eating clams and zooplankton in the Arctic in 2019? Well, we see 22 from clams to walruses, which if you look up in the human area there, that's a dose that would have an effect in humans. If you look at the bowhead dose through zooplankton, it's much lower. So this is the type of information that we're getting and that we want to be using to try to figure out if there are going to be health impacts to wildlife. So, question?
Yeah, no, I really want to get this right for my brain. Can you go back to that table, the one that has all those? So for the humans, they're all in red. Were they all lethal, or was it just the high number that was lethal, the 230 to 411? Or were all of those?
No,
Those were like maybe you were sick or maybe had diarrhea or
the
throwing
first
up.
one, 2.1 was lowest observable effect, so sick. The 21 was lowest dose to cause illness, so sick. The high one there was definitely lethal. However, the last one there, 1 to 2, that was also lethal.
Oh, all right.
So that's kind of what I'm getting at. And the reason there's variability all around that is because these aren't laboratory studies. Those are actually what these people ate.
Okay.
So that's all we have to go with. Yep. Okay, so what do we know? We know that saxotoxin and paralytic shellfish toxins are present in Arctic food webs. Walruses and bowhead whales are exposed to these toxins through their diet. And now we know from this 2019 data set that estimated doses to bowhead whales suggest low exposure in bowheads, probably not harmful to the whales, at least in terms of acute toxicity. Now, I cannot say bowhead whales are never affected by paralytic shellfish toxins. That's not what I'm saying. But based on what we're showing from this particular year, it looks like the toxin concentrations are not likely to be harmful. We're going to be doing this for multiple years to sort of see if this is the standard thing or if it increases at different times, et cetera. However, for walruses, it does suggest there's a potential for higher exposures that could be harmful to walruses. And we're going to be obviously working together with the communities to try to determine if this is occurring or when it occurs. We know that toxin levels in food webs will be directly related to the size and duration of Alexandrium blooms and to cyst germination. And that, as I've already kind of shown you in the beginning, those two things are linked to warmer waters. Warmer waters are linked to larger blooms and higher cyst germination. Okay. Dean.
Yeah, I hate to bug you. One of the biggest benthic marine mammal grazers is actually in the area where you're showing in the Chukchi up to Ledger Bay are gray whales. Do you have any information on gray whales?
We
Because-
have, okay, go ahead.
Because they're plowing through this sediment laden with cysts, which carry toxins, and laden with clams. And I was just curious. So
No, that's a really good question. From this particular study in 2019 that I'm showing you, I don't have a set of gray whales, but as Gay sort of mentioned, I also run the Wildlife Algal Toxins Research and Response Network, and we do get gray whales. And actually we have a lot of gray whale samples from the gray whale UME, unusual mortality event, that we're looking at. And so that is a very good point. Gray whales, mudsuckers can absolutely be exposed. I don't have data for you today on that, but that is something that we will be looking at.
Do you have, this is Gay, do you have gray whale samples from the northern Bering Strait Chukchi? Because you're looking
I would
for
have to-
feces.
I'd really have to go back and look. We have many, many gray whales. There's been a long sort of standing interest in the gray whales for that. I'm sorry, I just don't have that data
No,
ready.
no, that's fine. So I'm just thinking in the Bering Strait region, sometimes there's access to freshly dead animals. It would be helpful
Okay,
to your study. Go ahead, caller.
Richmond had a question too.
Go ahead.
Minke whales, didn't study on minke whales, did you?
Not
We-
yet.
No,
Oh,
but,
go ahead, I'm
but I know what you're
wrong.
thinking. We did measure a minke whale before.
But have you included them in this study? Not just one, but I mean, are they,
Right.
would you take,
No.
if people were to get a hold of fresh minke intestine or fresh gray whale intestine, would you want that for your study? Would that be helpful?
Yes, and we've had that long-standing relationship. I love to get samples. And so any samples that you get that you want to send to us, we can do. And that's what we do, especially large whales. We're very interested in large whales.
Did that answer the question about the minke whale? I kind of cut you off. Sorry about that.
We don't have a lot of data for minke
Yeah.
whales.
Any other question? And we have other
Thank you.
callers on. You're welcome. We have other callers on, so don't be shy if you're a caller with a question, just speak right up.
They eat fish. And there's also killer whales that eat the seals and the fish.
All right, I'm gonna put,
So we get all those
if
back.
there's no other question from callers, I'm gonna
Okay.
put on mute, and that would be continue on.
Okay.
Speak up, callers, anytime.
Yeah. Okay. So this slide is kind of just says, hey, what we don't know, but want to know. And so I just kind of was thinking things that people might be wanting to know or interested in. So wouldn't it be great? We want to know, when will there be dangerous toxic blooms? When exactly? Well, we can't answer that, but we can start to develop models over multiple years of data that can start to make predictions about when it's more likely to have blooms. So that's something that we're working on, particularly with Don Anderson and his team at WHOI. Where will they occur? We're starting to get these maps and we can start to make those conclusions on where they're likely to be. How toxic will the food web get? Well, I've just shown you the 1 year of sampling. We're going to be doing this over multiple years to sort of get at how much toxin there is in the food web under different bloom scenarios, different years, different conditions. The last question on this sheet, when will it be safe to eat shellfish? Of course, that's the $1 million question that everyone wants to know. And unfortunately, it's very unsatisfying. We cannot answer that question as a research team. This is all under the same guidelines that the state gives for seafood safety. Those guidelines need to be followed for protection for paralytic shellfish poisoning. And this applies to clams harvested from the guts of walruses. That's something I am just sharing with you. Now that we know there are clams out there that are above regulatory limits in walrus feeding grounds, this is now a concern. And so if you look at the website from the state, it again is very unsatisfying for subsistence harvest communities, because basically it's saying how to avoid paralytic shellfish poisoning. Don't eat shellfish harvested from untested beaches. You can eat shellfish that's commercially sold because it's tested. Again, very unsatisfying for subsistence harvest communities. I wish that I had better information or a good answer for that. Oops. Okay, yeah. But what I can do that I think will be of interest, or what we can do with this data that will be interest to your communities, is what parts of marine mammals will contain saxitoxin. So we know that walruses and bowhead whales and other marine mammals are important subsistence resources for food and cultural practices. So can we determine toxin levels throughout, for example, the walrus, which we know has pretty high exposure? So I wanted to share with you just what the data that we have right now, what parts of marine mammals will contain saxitoxin. If you are following along in your handbook, this is the slide that has a table on it that says walrus ID, location, year, feces, muscle, blubber. So if you look at this table, I wanted to share this data with you because these are the 13 walruses from 2019. The feces of these animals all contained saxitoxin. The two at the top are the hottest, and those are close to regulatory limits. We were also able to get muscle and blubber from each of these animals. And what you can see is that saxitoxin was not detected in muscle and blubber, even when it was relatively high in the feces. This is the first sort of set of data we have. We're going to continue to do this with you so that we can keep track and get enough data to where we can, really have a good understanding of, will there ever be toxin levels that are dangerous in other parts of walruses for consumption. So in our ongoing walrus studies, we are now getting other tissues. So we get feces, intestine, liver, kidney, heart, brain, muscle, and blubber. We're collecting these samples. We're analyzing them for toxins. We need to get enough data from animals that are highly exposed to be able to answer this question. So when an animal eats a lot of toxin, are you going to find dangerous levels in the liver, kidney, heart, brain, muscle, blubber, etc.? That's the data that we want to share with you as we get it. And we're working on that now. Now, I'm going to show you this figure here. So if you're looking in your handbook, this says ongoing walrus studies. There's a picture of a walrus, and then there's a graph there. What I want to show you on this graph is the first set of dots on the graph above 2019. There's a 13 written there. That 13 is, those are 13 walruses, fecal samples collected in 2019. All of them had toxin. 2 of them were close to the regulatory limit. No toxin was detected in muscle and blubber. In 2021, we got another 23 walrus samples, fecal samples. And what you see in 2021 is only 61% of the animals had toxin present. So many of them were below or no toxin or below detection limit, and they were all low. So we did get the suite of tissues from these animals. We are analyzing them and looking at them, but they are not great candidates to answer the question because they didn't appear to have a very high exposure in 2021. So again, these are the kind of data that we're going to be getting over multiple years that will help us develop these models that tell us when they're exposed and how much and when you see health impacts. So one sort of last thing, we tried to rush and get a bunch of data together just because we had this opportunity to share it with you. Oh, we have a quick question, Mary Jean.
Yeah, no, go ahead, Kathy. I'll come back to it.
Okay, okay. So what I'm showing here is saxitoxin in clams over 3 years. Now, in 2019, we got out there and we got to sample everywhere in the whole food web. In 2020 and 2021, we did not get to do that because of COVID issues. We did get some clams though, and that's what I'm sharing here. We didn't get as many sites, but we did get some. So in 2019, you can see there are, if you're looking at your pamphlet, it says saxitoxin in clams over 3 years. In 2019, there were 3 locations with toxin levels above the regulatory limit. In 2020, there were also locations where clams were above the regulatory limit, again in the Bering Strait and up near Utqiagvik. In 2021, there is much less toxin observed in the clams. Now, there's some caveats around this in that these samples were taken a bit later. There's not as many, et cetera. So I just wanted to show you the potential that we are going to have variability between years, and we're going to be able to determine what kind of factors are affecting that variability. Or in other words, what are the environmental conditions where we see more toxins and bigger blooms, or less toxins and less blooms. And just to kind of finalize here, in 2019, we see high toxin levels in clams. We see walruses, 100% of the walruses have toxin in the feces. In 2020, we didn't get any walrus samples, but in 2021, we got walrus samples, a lot of walrus samples, low levels of toxin or no toxin, and less toxin in the clams or in their prey. So to sort of wrap this all up, and then I'm going to just be so excited to answer all these questions. What are we doing? We're tracking harmful algal toxins in Arctic food webs. Why are we doing it? Algal toxins are an increasing threat to wildlife, we think, due to climate change because of the warming ocean waters and the potential for larger, more frequent blooms and higher cyst germination. What have we found so far? Massive cyst beds, and that was presented by Don Anderson again a few weeks ago in his Strait Science seminar. Evidence for more frequent and toxic blooms, toxins throughout the food web, which we've just shown here. And this is the first time we've been able to make realistic estimates of toxin doses to marine mammals. How can we use this data? And this is the thing that's the most exciting, is we want to be able to develop a model for predicting when and where wildlife will be impacted. Under what environmental conditions do we get more blooms? Under what bloom conditions do we get toxin levels in the food web that are high enough to impact marine wildlife? So that's basically the gist of what we're doing. This is a large scale EcoHab grant. That's who's funding this, but it's just got so many collaborators. And this picture, this set of these amazing, awesome people here is just part of the people involved. These were the folks who were involved in getting samples in 2019. As I said, this is funded by NOAA and the EcoHab program. And here's some of the co-investigators listed here. And then finally, the most important part of this project, the most important team of this for teamwork group. And if you have this pamphlet, it's the very last, go to the very end, where the value of this project is being able to work with the communities in the Arctic to learn more about the environment. And we couldn't do this without that contribution and sort of the help of sampling of the marine mammals and the other resources. So our promise to you is that we will be sharing this data with you. We want to share this as we get it and work together to see what's happening in the Arctic. So that is all I have, and I'm happy to take some questions if there are more.
All right. Well, thank you very much, Kathy. I hope you can hear me. It says I'm muted, but before we start taking questions, which I'm sure there will be, for those of you who are able to type in, please remember to give the speaker some love. This is not obviously not an easy thing to be a public speaker, much less for someone in Seattle on a different later time zone, and a tough. We have a lot of questions, and so thank you so much, Kathi, for letting us interrupt you and stuff as you go along. So a shout out from all of those who are not on the phone but on a computer. Feel free to give Kathi some love there for our Straight Science speakers, because it's not easy, honestly, having done it myself. So because callers get priority, are there any questions from the callers? Callers are number one.
There's a couple of questions about whether pits are being tested or will be tested. And on the bottom fish species, the halibut.
So on
That, yeah, so you're,
wait one, they're gonna give,
Gropins?
okay. Anything else?
Are you gonna be doing tests on halibut and
The rest of the pinnipeds and sculpins, I think. The bottom fish.
okay,
So
those are,
the question I think is, and stop me, Savunga, if I got this wrong, but the question is, are you going to be doing testing on sculpins, tunicates, halibut, other pinnipeds like the seals and sea lion?
Minky?
inky and sea peaches?
Yep. And
Gray
any of
whale.
the other
That's
whales?
Gray whale shall be.
okay.
The sea wolves, they consume.
So
Okay, yeah.
if
The
people hand
orcas
in
or the
samples,
killer whale.
So we don't have a plan to do sculpins and halibut. We can do some of those we don't have a way to get regular samples, but if we can do some of that, if you have samples you'd like to have tested. For the marine mammals, we definitely do.
If they could do that at the fish pond, they throw away the guts. They could just put the guts in a bag and send them out to Gay to get the halibut tested. Because for the total weight for commercial fishing, you have to take the gills and the organs out. So whoever catches them could just put them in a bag and then send it out to Gay.
Yep.
you guys are going to send me gut bags. Well, I'll do
Get
it.
the halibut tested.
If it helps get this question answered, you send me whatever you want. I'll try to get you guys a freezer or I gotta get that freezer.
I'll do it.
Good. Thank you.
Hi, Diamede.
Sounds good. Claire.
So Kathi,
Yep,
are you
that
willing?
was,
Are you willing, Kathi? If one sec, Diamede, if people like Savunga has a list of samples, if they're able to send in intestine from gray whale or minke, if for some reason they get one fresh, or any kind of the tunicates, are you willing to run those?
Yeah. So let me just say, we definitely can do any whales. We definitely want to do whales or any marine mammals. And someone asked about other pinnipeds. We do ice seals regularly. We do sea lions. So absolutely any samples from them. The fish samples, the halibut, etc. You get the guts. We can definitely analyze those. But I want to emphasize that this is for the research study looking at food web stuff. But it isn't for seafood safety in terms of guidance. We wouldn't be able to analyze them to say, hey, they're safe to eat or not. That's what I'm getting at.
Yeah, and because you're confined as
Well, we eat
researchers.
that.
Go ahead.
Well, we eat the halibut. We fish for it, and it's a bottom feeder.
Right. And so I think what Kathy's just saying is send it in, but don't expect a quick turnaround to say like, so maybe I know we have people from the Norton Sound Health Corporation on, and they are getting at with their seawater sampling program, eventually they're going to be able to do more of a human safety, sort of an awareness throughout the region regarding, which I shouldn't talk about their project, but there is another thing that's getting at kind of a warning system. What Kathy's saying here is that she's doing research, and by law, she cannot say she's looking at human health. So she could absolutely, she will look at everything you send her to add to her food model. So if you want to send halibut, which I hope you do, if there's by chance a fresh minke, a chunk of intestine down by the anus, send it in to me and I'll subsample it for her and get the unnock on its way. But she's just saying she's not going to turn it around real quick because of research, and she won't be a monitor for human health. All of that information, though, from the animals will go into this model, and we need all that for baseline understanding of what the heck is going on with this potentially emerging problem. So, so yes, the answer is yes, send it in, but she's got to be cautious.
And
about how she
while I have everyone on here, I'd like to say for the samples that are sent in, basically the information we need is the date that it's collected and the location that it's collected. And then it needs to be frozen as soon as possible.
and what it was.
Yeah.
I'm getting a bag of intestine.
Yeah. Definitely tell us date,
If it's a halibut or a sculpin or a
location,
salmon.
and then what it is. What's the animal?
So I will, when you go EVs on that, I need your coordinates of where you get the EVs.
If not, then they could too.
I know,
They're
and
closet.
anybody can. That's what I'm saying.
And it can
I'll
be a
need
rough
the
estimate.
Location.
It can be a rough estimate, Tiffany, but
was
coordinates would be great.
collected, and then I need to freeze it.
Yeah, I use coordinates for fishing. Yeah, coordinates.
Awesome. Yep, that would be super helpful. If you don't have your GPS, though, don't feel like don't send it in. I mean, if you know where you are, just sort of say you're 5 miles west of Savunga on this day, and this was a whatever. Gulpin.
Yeah, including with the orange.
Okay, thank you guys.
Yes, thank
Yep.
you.
Yeah, thank
Um,
you.
Gay,
Yep,
Gay, can you call me after you're done?
yep. It might be a minute. I'm going to ask Kathy to stay on the line for a sec after too, but I'll call you right when we're done.
All right, thank you.
You bet. And I just sent you an email as well. So there's, well, we'll tell that in a second. So there were other callers. I heard Diamond. Did you have any question for Kathy?
Yep. Well, yeah, but anyways, yeah, my question was why not collect the present species in the area, you know, where we're like, for my, yep. So, anyways, today I am monitoring, I am doing crab monitoring and also sample collections for harmful algal bloom or harmful algae. Anyways, I was thinking, you know, I can't, Kathy, I'm sorry, I cannot follow along with your slides. Diamond has really bad internet, so I tried, you know, to follow along the best I could.
Okay.
Gay and I were thinking, yeah, so Gay, she was going to send me slides, but, you know, the helicopter is not flying today. Anyway, and why not collect the present species, you know? I mean, why? I mean, I think it would be pretty awesome, you know, since while I'm doing this monitoring out here, you know, why not collect the present species in these plots, you know, like the clams and the worms? Of course, you know, it's frozen right now, you know, over the ice, the ocean is frozen, and, you know, the only way we can get it, and in my brain, I saw a Van Veen grab, and that's something I remember from the Sakuliak. I think there's a way, you know, collect the samples of clams and worms because, one, the bearded seal, which is another food that we harvest, you know, from our ocean, and, you know, they eat those things too, right?
Yep.
Yep. So I'm thinking, my question, and maybe Dean might know, where might I find a Van Veen grab?
Yeah.
And this might be a good time. That's a good question, Opik. Thank you. And thank you for the sampling that you're doing. For Kathi, can you explain how you're going to get clam and worm samples this summer from the Bering Strait and Chukchi?
Yeah. So, and I don't know, I guess I didn't really mention it, but we have a full cruise set up for this summer in 2022. We're going to be getting clams and worms from all those locations again, from multiple cruises. And OPIC, that's exactly how we do it. They do bottom grabs, bring it up, and we take clams and worms that way. So maybe we can have a follow-up, OPIC, and sort of see about the issue of a Van Veen grab, what kind of what you might need to be able to sample where you are if you wanted to get clams and worms in association with your water samples. I think that's a fantastic idea.
So, and I just want to
Yep,
mention
and then
because, oh, sorry, go ahead, Opik, I'm glad.
Anyways, Dean, if I know you're listening, I know you're there, and hello, by the way. But Dean, I remember we used the Van Veen grab about a mile, maybe about a mile or a half a mile from our village. No, about a mile. We dropped the Van Veen grab and we pulled up clams and worms and other invertebrates. But I remember Dean was the one that was going to sample them, right, Dean? Or I don't think you were doing the sampling or the testing on the boat, but having them sent out to be tested.
Oh, but good to have you online here. I was on that cruise, and those were the benthic people that were actually sampling those worms and clams and things from the Van Veen. But as far as I know, none of those were ever run for toxins. But I just received a message a couple of days ago from Seth that DEC was kind of getting rid of some old equipment, and I think a Van Veen may be on there. So I will follow up and try to see if I can get that Van Veen for Diomede.
So
But
for those who,
oh, wow,
go ahead,
that would
Opik.
be, but the Van Veen, I mean, there has to be a smaller Van Veen grab for me to be able to use it.
Yeah,
I'll
I
need
don't.
equipment
I
to-
really
This
don't
might
know
be
what.
Dean, this might be a conversation between you and Opik for getting equipment out to Diomede, and I think that would be super.
Understood.
Is that something you guys can work
Okay.
out, Opik? Just how to send it and where
Okay,
it might be, that might be. I just think that'd be great.
Thank
And know that we do hear of all these research cruises, and we're glad they're coming back. And know that when people are talking about the bottom trawl survey or Sekuliak, a lot of the research that's going on those boats, they are contributing to Kathy's to this study. And that's how she's not in charge of all these research vessels, but they're out there doing their study, and everybody tries to play nice, and so they're contributing. Just like when you were saying with your grabs on other ships, people put aside, Kathi asked nice, and they say, oh, you want 2 clams? All right. But that's how she's able to network and make this work. And so when we think about what we see with these research vessels, sometimes they're doing multiple different favors for lots of people. And one of them is this one, which is paying off with this kind of information for the Bering Strait region. And I'm just grateful that Kathi, you are so well connected with all these researchers, and that the research community on these boats is being nice enough to store you some clams in the freezer until they get back. So, and thank you, Opik, because if you can get clams right from Diomede, gee whiz, that's fantastic. So
yeah. And how about since we harvest ugruk and seals, I know the tribe was collecting samples and paying, who was it? Fish and Game, was it, that they're collecting the seal samples from? I've never personally collected samples from the seals myself. The samples I did send was directly, the walrus that I sent to you, Gay.
Yep.
And I know stuff was taking samples, but you know, they were going to a different, a
Program.
different place.
Yep.
Yeah.
And
Yeah.
Kathy's working with them as well.
Yeah,
is
I was
my
going to say,
understanding, so.
OPIC, we get those samples from Fish and Game. And in fact, I think we had several hundred ice seals just delivered to our lab today or yesterday. Emily
Oh.
Bowers works in my lab, has received those to analyze them. So we are
Wow.
connected through Fish and Game for that.
And
Okay.
Peter, I see your hand is raised, but I want to make sure the callers get their, get a chance, because it's awful to be a caller on a Zoom call. You can't raise your hand, and you're being very patient. Thank you. Opik, any other thing from Diomede? You guys
not
got the
right
open
now.
water?
We do. In fact, yeah, both sides still remain open. I mean, just a further out than, as compared to the year before. But yep, the water, when the north wind blows, the south side goes out. The south wind blows, the north side goes out. But then, it all comes back. Just
Right.
won't freeze thick. And then the crabbing area, we are a little further south than we were last year for crabbing. And the ice is about maybe 4.5 feet thick.
Okay. Well, that's great. Thank you
Mhm.
for that report. Anything else from?
Nope.
Okay.
Nope.
Well, thank you very much. Great to hear you out there. Glad your phone's working,
Mhm.
honestly. And any other callers? We have several callers online. So if you have any questions, feel free to jump right in. And while people might be waiting for their questions, Peter, go ahead. You've got your hand up.
Thanks. That was hugely fascinating. I wanted to ask, because I'm hearing so many people taking samples, how hard, so a minute ago, I guess the question about halibut and you're not testing specifically for food safety issues, how hard would it be to test that essentially at home? Is it possible to get a small, I don't know what the testing mechanism is, but if people are talking about getting their own Van Bean grabs, I figured you could probably have a small localized testing system that maybe doesn't feed directly into your research, but has some kind of citizen science contribution to it, if that's viable. And then also, so that's one question, and then unrelated question, are you seeing kind of from water samples to zooplankton samples to clam samples to walrus samples to human samples, are you seeing any clear patterns in how the toxins move up the food web?
Okay. So let's start with the first question about testing, and that's you're hitting on a massive topic, because ideally each community could have a lab that tests for toxins for seafood safety for the foods that they're harvesting. It is not easy to do that. The analyses are, it's analytical chemistry, the equipment is expensive, the expertise is needed. There's been just a massive effort at trying to make quick tests that you could take in the field, and we're just not there. It's just not available to have a reliable, quick, easy field test, because you're talking about human safety. If there's a false negative, or whatever's not done correctly, then there are can be lethal consequences. So Peter, that is something that should be a goal for the future, but we are not there, and it is not that easy.
I just by coincidence lived with someone who worked for USDA a couple of was one of my roommates, and she was during the pandemic, she was doing PCR tests for other things at home, and she said if they gave me a monitor, I could run all of our PCR COVID tests at home. So I didn't know if it was perhaps quite that translatable, but
no, that's great about PCR, but the paralytic shellfish toxins are actually a suite of toxins, and detection is it's not that easy. So
Okay.
Dina, you wanted to comment on that?
Yeah, Peter, I would suggest that you contact Rachel Lee at the Norton Sound Health Corporation and give her all the support you can, because I truly believe that an active toxin lab in your region will facilitate a lot of answers to the entire region. We're talking out to St. Lawrence, we're talking Wales, we're talking Nome, we're talking that entire region. And I think any support you can give to that and any push you can give them to facilitating a lab that will analyze toxins will get you faster results than any of our scientific studies. The scientific studies are there. They're showing that there's a potential problem, but we just can't turn the information around fast enough. And I think a health corporation that is fed with volunteers from the entire region can do a much better job. So I would highly support, I would highly endorse the Norton Sound Health Corporation to do such things.
fascinating. Thank you. I'll reach out to a Rachel Lee.
Yep. Great, Dean.
All right. Any other, Peter, does that answer your questions?
It answered the first one. The second one was unrelated and kind of veered away, but it only touch on the second if there aren't other questions.
Okay. All right. Let me just check here. A new message came in. Okay. So I guess, does anyone else have questions? I know I have a question or two. And I would, hearing none, I guess I'll ask my questions. And that is, can we go back to the slide that has the walrus tissues with all the list of the suite of tissues? And these walruses were harvested near St. Lawrence Island. And that's with the U.S. Fish and Wildlife Service Walrus Harvest Monitoring Program. Not that one, the one that actually has a picture of a walrus and everything that people are taking.
Oh, I see.
There we go. And for those, if you're still on from St. Lawrence Island or Diomede, this is the slide. Sorry, Diomede. This is the slide that says ongoing walrus studies, and it lists feces, because that's what she needs. It also lists looking at intestine, liver, kidney, heart, brain, muscle, and blubber. And I just wanted to make sure that people saw that, and if there was any other edible part of the walrus that isn't on that list that might need to be on that list. I was thinking, I do know people do eat stomachs of the bearded seal, the actual stomach. I don't know how much of that goes on anymore, but if there's any other tissue that people from St. Lawrence Island, Diomede, Elam, if you're still on, I saw Elam was on. If there's any other part of the walrus that is consumed, but not on this list, either speak up now or text me or let me know. And Kathi would probably be willing to maybe expand the tissue testing. If that's not covering it all for what is potentially edible on a walrus, I think she would really want to know that.
Yes.
She could get at what it is that might be, because this particular study I'm really interested in, because yes, we have to be mindful that a walrus could be sick because he ate too much clams that had eaten too much because of the bad algae, because I guess the water's too warm and there's a bloom of bad algae at Alexandrium. This should have been called the hunt for Alexandrium. If we see if the clams are eating Alexandrium, then the walrus eats the clams, and then it's in his intestine or it's in his stomach or in his feces. This one's really interesting to me because he may be that way, but it'd be okay to consume, for people to consume the body parts. that is really important, I think.
What about the walrus breast
So that would be the mammary gland.
and baby walrus?
Baby walrus
Yes.
would be nursing, so not eating clam. And I guess yeah, a calf would be nursing, not eating the algae. But maybe the milk.
Yeah.
Yeah.
So,
What about the baby?
is that what you would call Kushni?
Yeah.
Okay.
Yeah, Kusni and Kasekok.
Yep. And so, and for Diamede, I think you're still on. That would be Mammon. If you
Yep.
want those, if you want that part, that's the mammary gland. So if you want that,
Yep.
would that be all right, Kathy? Because that actually would then answer for the calf. If there's any kind of transfer there in the milk.
Yes.
So
we
the calf,
use-
we don't really need a tissue from, I don't think, but if you want to add, would you be willing to add mammary gland?
Yes. Now we have to do the proper permitting. So for this year, we've already put that in and we've gotten our permission, and so we can do this. But I could, for the next year, we can add mammary gland, and that's a good idea. The another algal toxin that I've worked with more, domoic acid, which I'm not going to talk about too much, but we do know that toxin is transferred through from mom to fetus or mother to pup through milk, etc. So it is a good question.
And I'm going to put in the chat for everybody. There was it just came out today. It's a bulletin that Kathi and a bunch of other people put out, and I emailed it to you, Tiffany, and I will email it to you, OPIC. And actually, I'll email it to all the tribal offices in the region. It literally just came out today. They were actually still working on it a little bit. And it addresses some of the subsistence concerns. Specifically, I just put it in the chat box for those of you who might be interested to download it. So anyway, there's that. I'm losing my train of thought. We'll leave it at that.
Yeah, it's sort of a one-page easy summary of all of this information.
Well, and it gets right at the subsistence issue. So anyway,
Yeah.
if there's any other tissues, I'm glad that came up. And maybe the permit question can be, if people are willing to give that to the Fish and Wildlife Service this year, you may not be able to get it until next year, but maybe they can fast pace that request. And I think that's worthy of a call to Fish and Wildlife Service, if they can put that on the list, if that's what the tribe wants. And you guys can, Fish and Wildlife can talk through their channels about getting that. But if there's an opportunity to get it this year, boy, if you may not see it for a year, I think they can probably fast track that, but
Okay. Yeah, we can
yeah,
definitely
we can talk
about.
offline on that more. And then my other question is, if people are one thing, and that's what that handout gets at, some of the 24/7, know that in the state of Alaska, paralytic shellfish poisoning is something that people in the Gulf of Alaska and Southeast Alaska live with every day. And that is why in the Gulf of Alaska and Southeast Alaska, where water is warmer, they have a little bit more of this, a lot more of this Alexandrium, and they have more issues with other toxins. And that handout tells you, for the state of Alaska, we have, there are 24/7, 24 hours a day, 7 days a week. There are 3 registered nurses on call, and those numbers are on that sheet. So know that this is not, we are not the first ones to deal with a problem of this issue of what is harmful algal blooms, what is saxitoxin, what's paralytic shellfish poisoning. So Alaska knows this is a problem. People deal with it more often, tribally, especially down in Southeast, and also tourists and other residents and things like that. And so we got those numbers, and those numbers are also given out to the clinics, I believe, as well. So hopefully that will be helpful to you. My question to Kathi is, if we're out boating or we're at the beach, and Diamede had this happen, what are some of the behaviors that might be something to think about. Is there anything you can tell us, Kathi, if an animal may be dosed up hard with a lot of this saxitoxin that we wouldn't know is around, what should we look for in an animal perhaps? Especially something like a walrus or I don't know.
That's such a great
Mammal.
question. This toxin, as I was saying, blocks nerve function so that nerves cannot fire. So there's a paralysis type of effect is its major thing. So I would say that if you notice an animal that is paralyzed, cannot move, or super lethargic, or is approached and it doesn't do anything, that is potentially, now this needs to be defined. We don't have a definition right now of exactly what saxitoxin poisoning would look like in a walrus. The other toxin I mentioned earlier. Oh, go ahead.
Yeah, go ahead, Opa, because I think you did have something very unusual happen in 2017.
Yep, we did, and it was in June. Remember, Gay? It was June 20. Was it 2018 or 19?
It's
I can't remember the year, but anyway. No, I'm talking about no, not that one. It was the one after that. It was the one where this walrus came up. This walrus came up by the school, and we knew it was sick. The person who spotted it, he says it just came right up. It was fearless. It came up. It was so skinny, and it had like patches on its arm. You know, the skin was discolored. So underneath its flippers, everything, there was a discoloration.
Oh, I remember that one.
Yeah, I took a whole bunch.
That had physical damage. That one.
Yep. Well,
yeah.
Outside his body, and then So this walrus came all the way up to the beach, even though people were here, they shot it, and they knew not to take the meat or anything because it really looked sick, and it did look, when it was coming ashore, it was like drunk or something.
So can you tell us about the walrus that you were able to sample that did have a high level of saxitoxin? Do you remember that one in 2017 that the guys called in?
In August? Yep.
Yeah.
Yeah.
That big bull?
I don't think it was that week. Oh yeah, there was the walrus. So this sick walrus was surrounded by other walrus, a couple of other walrus. And so those walrus were trying to help that sick walrus to shore. And so it did climb ashore, and the other walruses, they kind of took off after it got close enough to climb. I remember that, and it was shot right there.
And then you sampled it.
Yep.
And it turned out that one had quite a bit of. Kathi, you should answer that, and I'll put the link to that one in the chat box, and I'll send that also to the tribes.
Well, this conversation is just reminding me in terms of other things to look for based on what you just said, Opa. Another sort of thing when it comes to acute or high level exposure to algal toxins as the cause of death, the direct cause of death, it's more likely that the animal will actually kind of look healthy. It won't be starved. There won't be other damage to it. If it's sort of a normal, healthy animal swimming around getting too much toxin, then it will have impact. So this is just an interesting thing. Obviously that walrus that you saw that had all those impacts, there was something wrong. But in terms of algal toxins, we normally see kind of the death of what look almost like regular animals, and there's no real explanation as to why it died. So it's more likely that you're going to see a healthier looking animal, but behaving differently. And another toxin that is out in the environment, but not really at high levels where you are, but we see it down in California and Washington and Oregon, is the domoic acid, and that's an excitotoxin. It overstimulates the nerves, and in that case we see seizures in marine mammals. So with that toxin, there's kind of like a seizure activity. With Saxotoxin, I would expect more like what you saw with a lack of activity or not able to move.
Tired.
It probably would look tired.
So you're testing for biotoxins
Yeah.
in those? Okay.
Yes, absolutely. Yeah.
So,
So-
yeah, so that walrus, it was probably, it probably, I bet you it's still, you know, because the 3 target species of the algal bloom, you know, I don't have that sheet right in front of me. One of my workers took my book, but the 2 other species, the Dinophyces and the Pseudonychia, algal bloom. So I just I'm just curious because when we did our water sampling, you know, with the just to do identification to look for the 3 species, we did find Dinophyces 5 times. You know, and I'm pretty sure there were plenty more, but you know, with the slides that we're reviewing, we're seeing Dinophyces. That was the first species that we found.
Wow.
So that was with the Norton Sound Health Corporation, I believe. That's their sampling. Is that correct?
No. My project, Norton Sound, had nothing to do with it.
Okay.
My project, I invited Rachel Lee to come and train with us. You know, since she was going to do the same kind of project, pretty much.
Okay.
In Nome.
Yep.
Hey, since I'm in 2 people, a trainer. Why don't you come? So she joined in. Yeah.
Good. All right. Well, thank you very much, Opik. And thank you. That was, I think you were one of the 2017, that bull walrus being reported as so tired. And then when you were good enough and got those, the fecal samples sent in, and boom, that walrus was,
It had very high levels of saxitoxin.
very high levels. And he was still, and the other thing I wanted to mention is with all this sort of like, holy cow, all these scary stories, since, you know, this, what she's reporting on tonight, what Kathy's reporting on was 2019. And, you know, we haven't had any sickness from anybody eating native foods reported anyway at the hospital. So we don't know.
Are you also testing for, you know, besides I know you're focusing on saxitoxin tonight and Alexandrium. So the other 2 species, you know, and we were just talking about domoic acid. When you're doing these sampling with the walruses, are you also checking for domoic acid in these
Yes.
samples?
Yep. So every one of these samples that I showed tonight, we've also tested for domoic acid.
Okay.
Yep. And all of that information, that is not, I can just tell you the reason I'm not really presenting that is that those are lower levels. The bigger concern is saxitoxin. And that's why I was sharing this tonight. We do see domoic acid, but the levels are not alarming at this point.
Not unexpected. That's what that sounds like. So that's kind of good. Low levels. We like low levels. We don't like any of it, but we'll be happy with low levels. That word is good.
Just thinking, walrus that was sampled by the school, I bet you it had higher levels of domoic acid.
Well,
The one that was by the.
Yeah, so there's a little bit of a thing with domoic acid. So Opik, let me tell you this and see if you think my reasoning is good. Saxitoxin's regulatory limit is 80 micrograms per 100 grams shellfish. If you convert that to per gram, so it's 800 micrograms per gram. The regulatory limit for domoic acid is 20,000, I'm sorry, 800 nanograms per gram or 20,000. So the comparison is that saxitoxin is way more toxic than domoic acid. So you can't really directly compare concentrations. For example, 800 would be a dangerous level for saxitoxin, and 20,000 would be a dangerous level for domoic acid in terms of human health. So that's part of the reason I'm saying in terms of how toxic these toxins are, the concentrations, even though they might be measured higher, they're not more toxic. Does that make sense, Opik?
Sorry, I put you on mute. Kind of. I mean,
So
you know,
you might-
I could see some, but I'm just thinking, you know, I mean, you would, you know, when you're measuring for saxitoxin and you're measuring for domoic acid, you know, I'm just thinking in my mind that, you know, that walrus that was sick and had all the symptoms of it, you know, there could have been higher levels where he was, you know, whether he was, you know, swimming through it or, you know, where it was present in that body of water, you know?
So Opik, thank you. And that is the reason why it's always good in my mind, it's always good to sample, because now we won't be able to look back on that one. But if you get another one that is, you know, that other one you mentioned, it had all that like physical weird stuff on it. But any walrus that isn't correct, I would say get a sample of it, and from whatever we're able to physically get, Kathi will be able to get a number back. And so those kind of questions can get answered, because she is looking for
But she's
both.
okay. So if she's sampling it, I mean, Kathi, if you're sampling it, so this means that if you're sampling it and if you're seeing pseudonychia, that's the cause of domoic acid, right?
You are correct. Yes.
Yes.
And
So
we're
then maybe that walrus is eating more pseudonychia than Alexandrium. So I'm just saying there's more pseudonychia than Alexandrium, you know, present in that water, or that these shellfish ate more pseudonychia than Alexandrium.
And we'll know
because
that
here was
next. Oh, go ahead.
Around and pulled samples to look under the microscope, and we're finding more dinophyces than, you know, pseudonychia and Alexandrium, you know.
Right.
So
Yes.
wherever the
With
walrus
the
was,
sea-
it was
Yeah.
present.
I just want to reiterate how important the work you are doing is, because you're collecting seawater samples, and that is going to look at what's in the water. That's hugely important. And then what's going on with any of the samples that come in that people want tested. I mean, this is a food web study, so it's really important. And that's why I focus specifically on going back over these walrus tissues, both for human eating concerns, but know that if you do get another walrus and it gets sampled, Kathy's going to be looking for saxitoxin and the other one that you were talking about, pseudonychia.
Right.
And
just
so if that helps answer that.
can just follow up real quickly to sort of restate what I was saying, is that it takes less Alexandrium cells to be more dangerous than pseudonychia cells. You can have
I don't
more-
think that, anyway. Go ahead.
But anyway, let's talk off. We can discuss this later.
Yeah, people may have other
There's
questions,
so much-
but thank you very much, Opik. Lots of good talk there, lots of
Okay.
good questions.
I think I see Dean's hands on.
Yeah, I want to step away from marine mammals and just discuss the effects of saxotoxin on birds. When you're observing birds and they're on the beach and they're staggering around, acting drunk, or if you're at sea and their heads are drooping, these are indications that they're struggling probably with one of these toxins. And the birds typically get disoriented, they stop feeding, and that's when, you know, when we pick them up, everybody says, oh, they died of starvation, and that's because they went off their feed. So that's, if you're seeing marine mammals that are acting kind of odd, acting drunken, spinning around, or any of these unusual sights, those are the ones to report, because they're probably intoxicated with one of the toxins. And in reference to OPIC's concern about domoic acid. Domoic acid, the highest concentrations we've seen in the state so far have been down in Homer, in South Alaska, Southeast Alaska, and none of those concentrations of domoic acid toxins have been reported to be high enough to affect consumers. And so I would put domoic acid and the dinophysis toxins on the back burner and concentrate on Kathy's PSP levels, because I think they're much more dangerous, at least to the people consuming marine mammals, birds, and any tunicates, sea peaches, any of those types of things. And those are the things to watch for when the temperature levels are increasing. So, you know, I highly support the effects of collecting those animals for analysis. And again, I highly support any attempt that Norton Sound puts together to actually quickly evaluate toxin levels in some of these things, because I think it'll be a paramount concern to the entire region. And, you know, OPIC, I know you're concerned about these other things, but right now I'd be worried about the things that can kill you rather than things that can make you just sick.
So I just, thank you, Dean. Thank you, Dean. I just wanted to check on Kathy. She's been taking questions right and left here. Kathy, you all right with the question load? You hanging in there?
Yes, I think,
Okay.
Did Peter sign off?
He did not sign off. He just put his hand down. He
Oh.
had a question in the chat though, on the mother to pup question. How
Yeah.
measurably does the toxin move from water to zooplankton to walrus to humans? And I think that's
So that's the
the big
question,
question.
the original second question that you had, Peter, I remember now. And I wanted to tell you, that is exactly what we're doing with this project. That is the model that we want to develop. We are going to be developing a model that says, because we're taking all these samples, we're taking cell counts as well. For all these samples that I just showed you, we have the cell counts, we have other things. So yes, we want to be able to say, when the bloom density is this, toxin levels in zooplankton are this, toxin levels in clams are this, worms, etc. And toxin levels to these higher predators will be this, or doses will be this. So we're going to be modeling that. So then we'll be able to say in a year, well, if the bloom conditions are expected to be like this, then we know there's going to be impacts in wildlife or that kind of thing. That's what we're trying to do. So that's this kind of what we call a trophic transfer model. So we are going to be able to quantitatively model that, Peter. We don't have that information right now, but we're going to be incorporating cell densities, how much toxins there, how much we see in the different parts of the food web, how much things are eating. We're going to incorporate uptake and depuration rates for these organisms, etc. So that is exactly what we want to do.
And
Glad I understood it. Thanks.
sorry, Peter, step on you. Sorry. And OPIC, if you're still on the line, know that you actually got a shout out from one of the audience members who says to you, because you're a caller, appreciate the level of detail in your log, and I think recording behavior is useful. So people are hearing you, OPIC, and just wanted you to know the Zoom internet people are shouting out to you. So you got a kudo there. Thank you for speaking up. All right, with that, any other questions? Well, a lot of topics from one little algae. Who knew? I'm on the hunt for Alexandrium, and then there's these other ones that have their own other sets of problems, but the hunt for the Alexandrium is what I think I'm going to call this. And we're just so grateful.
I have one more question.
Go ahead.
One more question.
Hey, Gay, Gay,
Yeah.
Okay.
this is Tiffany. I have a question. Last summer, I was at Book and I was walking around the beach and there was a small rock and it didn't fly away, didn't do anything, and I just walked right up to it and picked it up. Do we send you samples of birds like that? It looked fine, but it just wasn't doing anything.
Right. Well, there is going to be, I just got a, so normally I would say yes, but but in May, we're going to have a straight science that's going to talk about avian influenza, which is another problem that may be coming our way, but we're unsure of that at this time. There is avian influenza we're having a little outbreak in the lower 48. It is affecting chicken farms and turkey farms, but there has been some wild birds in the flyways, even in the West, that some of the birds have avian influenza, which is tough for birds. And so there's a low chatter about it going on because it hasn't gone into a big deal yet. And so we'll know more in May. And I already called Fish and Wildlife and said, I've heard this low chatter. We need to hear about this in the region. So they are going to talk to us in May. So normally, yes, I would say absolutely. If you see some bird that is not acting right or it's, I can't really handle, well, if you send in a live bird, it will probably end up going to the bird TLC center in Anchorage, and we may get, if it's dead, it's easier to get samples from it, of course. If it's live, then there'll be a big effort to keep it alive, so rightly so. But I'll talk to you more about that, Tiffany, as the summer is upon us. So thank you for bringing that up. Sorry that doesn't help.
All right, thank you, Gay.
Yep. And then Diamede had another question. Go ahead.
I think I forgot, but so I'm thinking of,
Like
I know
me.
you're, again, I know you're focusing on Alexandrium, like you said, but then, when we're on the hunt for 3 target species, I'm just wondering, like these species, so they're probably, they're in the body, they're everywhere, right? So,
Yeah.
if they're present in the water, like if they were together, and they were consumed by a mammal, then it bioaccumulates, eaten by a fish and it bioaccumulates to the walrus.
Yeah.
Like, are there separate testing for different species, or, how can you tell if it's from Alexandrium, and then how can you, I don't know.
Yeah, so
I'm just thinking
we are testing, we are doing everything that I just showed you for Pseudonychia. We're not doing Dinophysis, but we are doing Pseudonychia. So we do have comprehensive data for domoic acid. And maybe that's something I'll be able to do in another seminar to sort of share that in a few months or so. But so yes, so you could rest assured there of your 3 target species you're talking about, we are comprehensively modeling and doing Alexandrium, Saxotoxin, and Pseudonychia and domoic acid. So and we know there are co-exposures. They're being exposed to both toxins all the time.
both are 3?
I don't have data on Dinophysis.
And
And
I think Opik and I were sort of on a little chat on the side, and it looks like maybe Kathi, if you could be in a call, not right now, but at a later time with Opik to maybe really auger in on some of this, because she is trained up and is doing a great job. It might be a longer conversation that would be really helpful, I think, for everybody, because she's really looking at a suite of stuff out there.
Yeah, sounds good.
Yeah. Would that work for you, Opik?
That works fine. Yep. That works great.
Awesome.
Thank you.
Awesome. All right. Well, Any other questions? Kathy, I want to thank you, and I thank the audience for staying with us, and I thank the audience for all the kudos for Kathy in the chat, because it is not easy, and this is a tough topic, and one that I'm trying to learn a lot about, as this is seemingly an emerging issue for the Bering Strait region. It's a problem for people in Southeast. They've learned how to live with it, and it's like everything else changing. So I'm trying to learn and be hip. And I'm grateful that Kathy, you are, and all the researchers are contributing, that the communities are contributing, and that you're taking the time here to tell us what you found. And it looks like, I can already tell you, we want you back. So come back to Strait Science, that's for sure.
Great.
We'll want to hear what you find. So with that, for next week, this is a 1-2. Next week on week from tonight at 6:30 will be Rick Toman. He'll be talking about the wild winter weather we have had, and it also give us a little preview into spring, what may happen. And for those of us in the Bering Strait region that are going to be extremely interested in ice conditions in the next month or two, I really encourage you to tune in next Thursday at 6:30, where Rick Toman's gonna, with the University of Alaska Fairbanks ACAP program, the Alaska Center for Climate Assessment and Policy. Hope I got that right. And stay tuned, because he's come on back. He's gonna give us what we went through this winter, why potentially, maybe some reasons as to why, even though it was cold all winter, why our ice is still so thin, and what we might expect this spring. And actually, after listening to Kathy's talk tonight about warming waters, it'll be even more interesting for me anyway to hear what Rick has to say. So please stay tuned, and we'll see you hopefully back next Thursday night. With that, thank you all for coming.